Hello, fellow neuroscientists. I'm pleased to announce that my organization, the Brain Preservation Foundation, is starting a new series of prizes called the Aspirational Neuroscience Prizes, designed to highlight and honor recent research regarding how long-term learning and memory are encoded in the brain at the structural and molecular levels. We will also be hosting a related social and panel discussion at the Society for Neuroscience conference. If you are a neuroscientist who is well versed in the learning and memory literature, please consider offering up nominations or assisting us with the review process. Please contact me personally at the email address listed here and use the hashtag Aspirational Neuroscience to follow nominations on Twitter. A complete overview of the prize is available on our website. Here is just a taste of the types of research that are being considered for nominations. Behavioral research, identifying distinct types of memories and memory systems. Structural correlates of long-term potentiation, as viewed by light and electron microscopy. Glutamate uncaging experiments that probe structure-function correlations. Experiments tracking molecules at synapses during LTP. Transgenic engram tagging and optogenetic reactivation experiments. Experiments using fluorescent protein reconstitution across synaptic partners to identify engram connectivity. Synaptic ensemble tagging and photomanipulation experiments. Experiments performing calcium imaging of the receptive field properties of single dendritic spines during learning. Experiments which use holographic manipulation of cortical ensembles via optogenetics. EM connectomic experiments analyzing synapse size correlations. EM connectomic reconstructions of neural circuits with known functional properties. Experiments exploring non-synaptic memory mechanisms. And many more. As you can see from this list, it is a truly fantastic time to be a neuroscientist studying the physical basis of learning and memory. We have an awesomely powerful set of experimental tools to test existing theories with. Let me say a few words about why we think the neuroscience community needs an aspirational prize. The human brain is the most complex system ever studied. The field of neuroscience has labored intensely for more than a century in its effort to understand the brain's functioning and is only beginning to unravel the full story. It is reasonable to expect that another century or more of increasingly well-funded research will be necessary before neuroscience can reach its ultimate goal, that of obtaining a comprehensive computational understanding of the brain. A reasonable question to ask is why humanity should continue to devote increasingly larger resources for so long toward such a goal. This question is especially poignant given that many in the general public mistakenly believe that we have learned little about how the brain works, and many believe that it is impossible to obtain a computational understanding of the brain. The Aspirational Neuroscience Prizes are designed to counter such pessimism, replacing it with an understanding of just how far we have already come in the journey. Hopefully, in the process, the prizes will also spark a spirited debate regarding the long-term benefits that eventual success might provide humanity. We at the Brain Preservation Foundation are motivated by one key assumption, that the neuroscience community will eventually succeed. We understand that it will take many decades, perhaps centuries, of diligent work, but we firmly believe that success is inevitable, and that from that success will flow the technologies to scan human brains at the connectomic and molecular levels, reading out their encoded memories with sufficient fidelity to allow for mind uploading. I know this sounds like science fiction, and it is science fiction, since we are nowhere close to achieving this today. But it seems to us reasonable to plan for this eventual success. Specifically, it seems to us reasonable to ask the following question. Does the neuroscience community now have in its toolbox the ability to preserve, for an indefinite time period, an entire human brain with such fidelity that its long-term memories remain, at least in principle, decodable by far future technology? If the answer to this question is yes, then such a brain preservation technique may act as a bridge that terminal patients could use to reach that future mind uploading technology. As you may know, a few years ago, our organization ran the Skeptical Brain Preservation Challenge Prize, which challenged the cryonics community to demonstrate that their methods could preserve at least the structural connectivity of the brain. Perhaps unsurprisingly, they failed to demonstrate even this minimal level of preservation. But it is important to understand that our challenge prize was eventually won by a team of scientists which simply modified a standard technique used by neuroscientists, glutaraldehyde perfusion fixation, making it compatible with long-term cryostorage. This technique, published under the name aldehyde-stabilized cryopreservation, was demonstrated to preserve whole pig brains, yielding textbook-quality electron micrographs of connectivity and ultrastructure. And the technique should also be preserving the locations and identities of a wide range of biomolecules crucial to neuronal function, including ion channels and receptor proteins. From our reading of the learning and memory literature, such a preservation technique seems sufficient to cover all the existing theories of how long-term memories are encoded in the brain. But this question should really be debated by the entire neuroscience community. Only the neuroscience community has the expertise needed to address this question adequately. The Aspirational Neuroscience Prizes and associated events are our way of starting such a dialogue and of focusing it on the relevant science, namely, how are long-term learning and memory encoded physically in the brain? Even if you vehemently disagree with the prospect of medical brain preservation, I hope you, as a neuroscientist, can at least appreciate that the question of how long-term memories are encoded physically in the brain is a scientifically tractable one, and one that is fundamental to our understanding of the brain. And I hope you will join us in honoring some of the best experiments that have recently been carried out to probe this question. For more information on the prize and nominations, see our website, brainpreservation.org, and follow the prize on Twitter with the hashtag Aspirational Neuroscience. Thank you.